Okay, hi everyone. My name is John Awaya. I'm a faculty here in the biology department. I primarily teach a lot of the microbiology courses, but I'm also kind of partly in charge of the genomics kind of facility, core facility here. We have a lot of these kind of instruments in this facility that's open to all the students, especially for the TCBS students that want to get hands-on work that's going to be more molecular based. Okay, so inside of this lab, everything that you might see around here is everything that's supposed to be really, really small and definitely really, really clean work. So we don't ever want to bring in anything in here that's going to be technically kind of dirty. So it's always going to be kind of more cleaner things. We're going to work a lot more with DNA and RNA work. So if that's something that you're really interested in, you're going to find out that a lot of the machines in here and a lot of the instruments are going to be right up your alley. Okay? So for example, when you're looking at something like this, I know this is backwards right now, but this is definitely the ion torrent, which is for next generation sequencing. Okay, so maybe you may hear about it and read about it, but you have an opportunity to even try it yourself. You know, if you have samples that might have to do something along the line of metagenomics. Okay, it comes in parts, so it's not like just one main machine. It kind of has all different parts to it. But another major piece of instrument I think gets a lot of use for everybody is going to be the real-time, the RT-PCR, the real-time PCR. So this is oftentimes for RNA work. If you got to do RNA work, it gets a little tricky because it has to be really clean. So we have kind of a clean hood that allows you to do all that work, and you want to make sure everything kind of stays in there when you're doing that kind of thing. And then from there, you can process it and go straight into your machine work. Water, filtration, refrigeration, and if you ever need to spin down anything cold like doing RNA work or your DNA work, we definitely have refrigerated centrifuges as well. And then the other big ticket item that a lot of students will use is a lot of this area. And this is going to be for measuring concentrations of DNA as well. Okay, so if you want to measure how concentrated your work is, this is something that you're going to probably use on a regular basis. So we oftentimes have a lot of students come in, they might just prep their samples, take their measurements, and then go do whatever else they want to do. Okay. The big last item that oftentimes kind of gets put to the side is because, you know, it doesn't get much use anymore, but this is definitely a sequencer. Okay, so a genetic sequencer that you want to do DNA sampling and then you want to sequence your samples. Well, this machine can do it, but the unfortunate thing is it costs quite a bit to keep running with small amounts of samples. So anytime we do run it, we're going to need a lot of them, you know, 100 samples to even more than that to make it kind of cost efficient. So sometimes a lot of students, if they have only like one or two things, they'll definitely send it out to UH Manoa or to another facility that you can get a better price with. But if this is something, again, that you're interested in learning about or doing, we can definitely help out and do things. So as of right now, it's getting limited use. I think we can still only have maybe 2 people in here at the most at a given time. I would say maybe only 1 person comes in here a day at the most. But it's welcome to everybody. The one thing I just make sure that you do is definitely clear it with me or have your PI talk to me so we can set up some type of schedule for you to be working in here. All right? So if you guys have any questions, definitely email me. If not, stay safe, and I'll talk to you guys later.